A very good morning to all of you. Today I am going to discuss about application of monoclonal antibody. How can you get the monoclonal antibody? Through hybridoma technology. So today topic is application of monoclonal antibody and stem cell technology. Now I am going to tell about the application of monoclonal antibody, that is the production of therapeutic antibody. You must listen. It must come your exam. It must come in your exam that what is OKT3? OKT3, it is a product of monoclonal antibody. It is a product of monoclonal antibody that is used during kidney transplantation. It is a immunosuppressive drug. It is a immunosuppressive drug. OKT3, it is a immunosuppressive drug that decrease the recognition power of T helper cell. How T helper cell getting the recognition power, whether it is a part of your body or not part of the body. So if you observe the T cell, on the head region of the T cell, there is a CD3 protein. So just CD3 protein act as a camera, means which one is going in your inside your body and which material is harmful and it is going to integrate in your body. So generally it will reject by that property due to presence of CD3 protein present on T helper cell. So what is the function of OKT3 monoclonal antibody? That will mask. Suppose your head is going to cover with black color cloths. Can you recognize your mother without without listening her voice? You cannot recognize. Okay. In the same way, T helper cell is responsible for recognizing the any body part, whether it is the part of your body or not part of the body. So how it is going to recognize? Due to presence of CD3 protein. So Pratibha, you have done the mistake. All of you have done the mistake. I have told you that write down the therapeutic monoclonal antibody. Then you have written CD3. It is not a CD3. It is a OKT3 that is used during kidney transplantation. CD3 is a protein present on where? Present on T helper cell. That due to which your body is going to recognize it is a part of your body, or it is this is the kidney of your parents or your brother or your sister. So the dose inject in the form of injection is going to enter in your body, that will reduce the recognition power of your body. And at the before the doing the kidney transplantation, if you have problem in your kidney, so then need of one fresh kidney. So before that surgery, they need of injection of OKT3 so that it can easily join. Otherwise, your body will reject. No, no blood relation will come helpful in that situation. Your body will reject. This is not a part of our body, my body. Okay, due to presence of CD3 protein present on T helper cell. Are you are you getting my points or not? What is the function of OKT3? What is the function of OKT3? It is clear or not? It will come in your CBSE exam. That's why I'm telling you. Next is therapeutic monoclonal antibody, that is herceptin. You know, this herceptin, it will deactivate the receptor. This herceptin, that is a tros trastuzumab monoclonal antibody. Trastuzumab monoclonal antibody. So you always remember the therapeutic monoclonal antibody. One is OKT3 and one is herceptin. What is the name of that herceptin? It is clear or not? Name of herceptin is trastuzumab. Trastuzumab. It is a monoclonal antibody for therapy of early stage breast cancer. Early stage breast cancer. So that is helpful for treatment of breast cancer when it is in early stage. Okay. So what happens? Human epidermal growth factor receptor 2 positive. Okay. So that is going to join with the cell surface receptor that help to grow and multiply. Okay. So when this monoclonal antibody is going to join with the uh, cell surface receptor, it will stop the multiplication of the cell present in the breast. What happens? When this herceptin, that is the monoclonal antibody, is going to join with the cell surface receptor, then what happens? It will stop the rapid multiplication of the cell present in the breast of the any lady who is going to suffer from breast cancer. Okay. So this will impair the growth of the breast cancer, means stop the growth of breast cancer. So all of you must listen carefully. What are the two therapeutic monoclonal antibody? One is OKT3 and one is Herceptin, that is used for treatment of breast cancer. How it is helpful? It is joined with the receptor. It is joined with the receptor where after receiving the signals for the growth of cancerous cell, instead of that, herceptin molecule, that is the monoclonal antibody, is going to join and it will prevent the growth of the cell of the breast cell. That is lady suffering from breast cancer. Okay, it is clear to all of you or not? Yes, 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 yeah, no, yes. So I have told you so many times who had discovered the monoclonal antibody. Who had discovered? Caesar Milestone and George Kohler, and got a Nobel Prize in 1975. Yes, they are the Nobel Prize winner, and this is the hybridoma technology. And I have told so many times, chemical used for fusion of two cells, polyethylene glycol and calcium ion. And in which medium you are going to select the hybrid cell? In which medium you are going to select the hybrid cell? Anyone? Anyone can tell me which in which medium you are going to select the hybrid cell? So this question must come in 5 marks in your coming competitive exam or in your CBSE exam. That's why I'm repeating these things again and again. So this is the product. This is the monoclonal antibody, the product of this hybridoma technology, and it is very helpful technique for production of monoclonal antibody for treatment of different type of disease and to develop the different type of diagnostic kits. So you are not telling any word, you are not replying. That's why I'm very sad of your batch, from your batch. So please reply yes, yeah, no. Otherwise, it is very problematic for me. Okay. Now come to STM cell technology. What is STM cell? A stem cell, it has a self renewal capacity, differentiation capacity. What mean by self renewal? Means one nandini, from one nandini cell you can make other nandini cell, and whole nandini will develop from one nandini. So that is the clone. That is the ability to divide, multiply, differentiate and develop whole individually. That is the meaning of that. So stem cell have capacity to divide, multiply and self renewal, means that is the capability to produce same like itself. This is the mystery. Okay, so this is the mystery in that particular cell. All cell have no such capacity. So just think, just imagine, you were a single zygote. You were a single zygote when you were a single zygote 16 years ago, when the male and female gamete form a zygote. then that zygote forms somatic means embryo. So that embryo is a stem cell. That embryo is a stem cell. How? Means from the single zygotic cell, we are going to develop a whole individual. So this is the mystery. That is the characteristic of God. Means self renewal and differentiation. This is the property of a stem cell, and you find this characteristic feature only in zygotic embryo. Okay, so zygotic embryo, that is the totipotent. So can you tell me what do you mean by totipotent? What do you mean by pluripotent? What do you mean by multipotent? What do you mean by unipotent? Anyone? What do you mean by totipotent? Ability of cell to divide, multiply, and regenerate whole individual. Means one cell is responsible for developing the whole individual. Means zygote was one of the diploid cell. From that cell, you were developed 17 year ago, 15 year ago. Suppose your age is 15 year or 16 year, like that. So in your mother womb, you were only a single zygote. So just see what is the mystery in that stem cell. That is the zygotic cell. That is the mystery. Means that is going to differentiate. That is going to self renewal. Means what? Just try to know self renewal means capacity to develop like themselves. And differentiation means that division of labor. Division of labor means all the cell going to differentiate. From a zygotic cell, there is a formation of nerve cell, there is a formation of muscle cell, there is a formation of bone cell, there is a formation of cornea, there is a formation of elementary epithelial cell. Means all the type of cell is going to differentiate. In earlier it was only a single cell, and then due to repeated division and cleavage, there is a zygotic embryo that develop into blastula, gastrula, and then ectoderm, mesoderm, endoderm. I think you have read these things in zoology, botany, means biology. So this is the mystery. Means we have all the body part, and these all are developed from a single cell, that is the zygotic cell. So scientists have studied that things, these things on the in the animal cell culture lab. Okay, so just see this self renewal capacity and differentiation capacity only present in the embryonic stem cell. So there are two type of embryonic stem cell. One is stem cell. One is embryonic stem cell. One is adult stem cell. So embryonic stem cell, it is the totipotent. A simple in the simple term we can I'm going to explain the totipotency means ability of a single cell to develop a whole individual. Suppose a zygote cell develop into a whole human body, human baby. That is the meaning of that totipotency. Means you can get a 1,000 plant from a single leaf. Means each leaf is going to develop into whole plant. That is the meaning of that. So such type of property generally more in case of plant than animal. Okay. So just try to understand the different terminology regarding the stem cell. Is just listen. There are two type of stem cell. One is embryonic stem cell and other is adult stem cell. Adult stem cell have some limitation. Means they have no such capacity to develop into whole individual. Only they will develop a certain part of your body. Okay, so that is multipotent or unipotent. Okay, so these things you have to understand. Means that property can be used for the treatment of different type of disease. Okay, so just see in this diagram. Okay, so this is a zygote. I am discussing this zygote, fertilized zygote, then morula stage. Just see, this is a morula after cleavage and division. Then this is the third one is blastula with inner mass and mass, and fourth is culture undifferentiated stem cell that is present. And then fifth is differentiated cell, that is the blood, neural, muscles. Means they were their parental cell was only one, that was the zygotic cell. So from that cell there is a differentiation, differentiation, and that's one form one out of the this number of the cell, one will form, one have formed the blood, one have formed the neural cell, and one have formed the muscle cell, and one have formed the bone cell also. So these are the differentiation, that is the mystery. So this type of property only present in that stem cell. Just see in this diagram. Okay. So just see in the bone marrow. Bone marrow also have a stem cell. So just formation of RBC occur from the bone marrow cell. Okay. So try to know this. अंडरस्टैंड दिस डायग्राम क्या है इसमें कि कल्टीवेशन ऑफ adult stem cell from the bone marrow and their differentiation into specialized cell means you can multiply that stem cell inside the animal cell culture lab after taking the bone marrow cell and put in the animal cell culture media or human cell culture media. Okay, and then we can differentiate into a specialized cell, and this thing is going on. Means suppose in any occasion, suppose your cornea affected with the crackers, then what happens, what you will do, you will not able to see. So scientists will take the skin of your body, and then it will they will culture that skin and modify that skin, means differentiate that skin into cornea, and then that cornea that can fit inside your eye, now you are able to see. So this is the mystery, this is the application of animal cell culture, and utility of that animal cell culture. So this is the meaning of that, means bone marrow have such capacity, bone marrow cell have such capacity, that is the self renewal capacity, differentiation capacity, that is present, and you can culture that and modify that cell into different type of cell, like neuron cell, like epithelial cell, like skeletal muscle cell, like means any type of cell you can differentiate from the one cell, that is the adult stem cell, that is bone marrow. So just try to learn these things, means the two property of a stem cell, one is differentiation, and second is self renewal capacity is only present in case of bone marrow cell and zygotic cell. Okay, so zygotic cell is advanced in term of totipotency or self renewal capacity or differentiation capacity in comparison to adult stem cell. So you have to learn these things, that such property that can be utilized for treatment of different type of disease. Yes, suppose your cornea is not working, then in that situation scientists will utilize that your skin cell, scientists will utilize your skin cell and differentiate into corneals. So this is the application part of animal cell culture technology. Okay, so everyone try to learn what do you mean by animal cell culture technology. Okay. Then what do you mean by stem cell culture technology. Okay, so just see, if you are observing this embryonic stem cell culture and its application, embryonic stem cell culture and its application, you can generate a chimeric mice, means inner cell mass. Generally embryonic stem cell line derived directly from the inner cell mass of the growing embryo without use of immortalizing or transforming agent. Means embryonic stem cell we can get from the growing embryo. So inner cell mass of embryo can be maintained in the cell culture in presence of irradiated fibroblast, that is the stem cell. So inner cell mass, If you are going to fuse the male gamete and female gamete after fertilization, zygote will form, and then the zygote will start multiplication cleavage. So in earlier stage, you are going to collect that cell. That cell is inner cell mass of that embryo. So that is the embryo. After zygote, there is development of embryo. From that embryo, you are collecting. You are collecting the cell, and you are going to develop that cell into the animal cell culture lab or human cell culture lab. Okay. So it retains the characteristic of founder cell even after prolonged culture of and extensive manipulation. Means what property it will retain? Self renewal capacity, differentiation capacity, and reintegrate fully into the embryogenesis if transferred. So it could be used to create a chimeric mice by taking embryonic stem cell from the black mice and implanted into embryo of albino mice. What is the meaning of that line? To try to know. Suppose you are doing the experiment and you want to transfer the characters from the black mice into the albino white mice. So this is the black mice, that is the you are growing the black mice after formation of embryo. Just after zygote development, you take out that embryonic stem cell from the earliest stage of embryo, and then you culture in that in the lab, animal cell culture lab. So मैडम अभी क्लास में थोड़ा बाद में बात. So just embryonic that is the you are doing the experiment that you want to transfer the character from the black mice into white mice. So what you have to do? First you have to develop the embryo. How can you develop the embryo? Just after fertilization, the development of zygote, and that zygote is going to develop into embryo. So that embryo, if it is in earlier stage, not in differentiated, more differentiated stage, you take out that, and the cell whatever you get from that embryo, you culture it in animal cell culture lab. Again, side by side, you are going to develop the embryo of albino mice and take out the cell of the embryonic stem cell of the albino mice, that is the embryo, and then you again you are culturing in the animal cell culture lab. Now you are going to mix the embryonic stem cell of black mice and the embryonic stem cell of albino white mice. After mixing, you develop the embryo in the inside the lab, and then you put that mixture of the cell of the embryonic cell of the black mice as well as the white mice into third one mice, that is not one, not the albino mice, not the black mice. Both are the parents, but you are not utilizing any one parents. So you are going to inject into the third one, that is known as surrogate. So inside that, when you are putting the trans embryo, now that embryonic stem cell that is known as trans embryonic cell. Why? Because that contain the both black mice cell embryonic stem cell as well as white mice embryonic stem cell. Just see, this is the white mice embryonic stem cell, this is the black mice embryonic stem cell. Now when you are going to develop that trans embryo in that third one, then there is a development of baby in that that mice. This is not the black albino mice, this is not the white albino mice, this is the third mice. So inside that mice, you are observing the whatever the offspring that contain the gene or character of both black mice as well as the white mice. So how can you confirm? You are observing the black strip color of the hair and white strip color of the hair on the surface of body of that mice. So that is the condition for creation of chimera. That is the condition for creation of chimeric mice. So just see, this is the chimeric mice. So how can you develop the chimeric mice? This is the application part. This is how can you mixing the character of the two different type of mice, whether one is the black mice and one is the albino or white mice. So first of all, you have to collect the embryonic stem cell. How can you collect the embryonic stem cell? Just after fertilization. Just after fertilization, there is a development of embryo in both black mice as well as albino mice. Then you take out the embryo from both the mice and culture the embryonic stem cell of the both of the mice, and then you transfer the some cell of the black mice of the embryonic stem cell of the black mice into with that one, that is the albino mice. Then that embryo is known as trans embryo. Now this trans embryo you are going to put inside the third one, that is third one, and that is the surrogate mother, where there is a development of chimeric. So all of you must try to know what do you mean by chimera. Means a mixture of two different character of two different mice, that is known as chimera. So yes, so all of you try to listen, how can you create a chimeric mice. Okay, so just go through that. What is the what is a stem cell? What is the property of a stem cell? How can you classify the stem cell? Whether it is the totipotent or multipotent or pluripotent or unipotent, and how can you develop the chimeric mice? So first go through that chapter and revise that chapter. If you have any question, you can ask me. If you have any question, you can ask me. So no question. Yes. Thank you.